everyone. Right now we're going to discuss the chapter 4 of microbiology and some certain topics that um, comprise the chapter 4. So, first is we're going to describe the characteristics used to classify viruses in terms of DNA versus RNA. So, viruses can be identified through the genome that they possess. So, what is a genome? Genome is the genetic material that the viruses possess. So, most viruses contain double stranded DNA or single stranded RNA, but some viruses have double stranded RNA or single stranded DNA. So, we have to note that some viruses contain RNA whereas others contain DNA, and the nucleic acid that they possess may either be single or double stranded. Within the host cell, single stranded positive sense RNA functions as the messenger RNA, whereas the single stranded negative sense RNA serves as a template for the production of messenger RNA. Some of the viruses possess an envelope, whereas others do not. So, number two, we're going to specify five properties of viruses that distinguish them from bacteria. So, first is in terms of size, the bacteria is larger than the viruses. Um, Second is in their cell wall, the bacteria contains peptidoglycan or lipopolysaccharide, whereas viruses do not have cell walls. They only have protein coat present. Okay. Next is the ribosomes. So bacteria have ribosomes, whereas viruses do not have. And next is the number of cells. So bacteria is a unicellular organism, meaning it is just a one cell organism. Whereas viruses, they are not considered as cells. Okay? Next is in terms of reproduction and their way of reproduction, the bacteria are able to reproduce on their own, whereas viruses, they need their host. They need the cell of their host or the host cell in order for them to reproduce. Number three is we're going to state at least three important viral diseases of humans. So, what are viral diseases? Viral diseases are extremely widespread infections caused by viruses, meaning these are diseases or infections that are caused by viruses. So, we have here chicken pox and flu, which is the influenza, and the human immunodeficiency virus or the HIV or AIDS. So, number four, we're going to discuss the differences between proviruses, viroids, and prions, and the diseases that they cause. So, first, what is a provirus? A provirus is a form of virus that is integrated into the genetic material of the host and by replicating with it can be transmitted from one cell generation to the next without causing lysis or cell death. So, a provirus does not directly make DNA copies of itself while being integrated into the host genome. Instead, it is passively replicated along with the host genome and passed on to the original cell's offspring. So, all the descendants of the infected cell will also bear proviruses in their genome. So, next, viroids and prions. Viroids are a bunch of single stranded RNA that attack the plant's metabolism that lead to several plant diseases and even death to some. And next is prions. Prions, on the other hand, are tiny proteins that cause deadly brain disorder or or neurological disorder to animals and humans or mammals. They are also called as mammals. Next is we're going to list various ways in which bacteria can be classified. So we have here some several ways in which a bacteria can be classified. Number one, classification of bacteria on the basis of shape. Number two, classification of bacteria on the basis of mode of nutrition. Number three, classification of bacteria on the basis of temperature requirement. Number four, classification of bacteria on the basis of oxygen requirement. Number five, classification of bacteria on the basis of pH of growth. Number six, classification of bacteria on the basis of osmotic pressure requirement. Number seven, classification of bacteria on the basis of number of flagella. Number eight is, and last, is the classification of bacteria on the basis of spore formation. So we're going to proceed to the next item. So number 8, we're going to state the 3 purposes of fixation. But first, what is fixation? Fixation is a chemical process that intends to lock the tissue, cells, and subcellular components as close as possible to its original status, even though such is virtually impossible through standard procedures. So, in general, fixation serves 3 purposes. Number 1, it kills the organisms. Number 2, it preserves their morphology or their shape. Number 3 is it anchors the smear to the slide. So next item, we're going to define some terms. The Diplococci, Streptococci, Staphylococci, Tetrad, Octad, Coccobacilli, Diplobacilli, Streptobacilli, and Pleomorphism. So first, Diplococci are cocci in pairs, meaning they are by 2 or, Next is Streptococci, which are the cocci in chains. Next, Staphylococci, cocci in clusters. So Tetrad is a packet of 4 cocci, while octad, from the word octa, is a packet of 8 cocci. So next, Coccobacilli are bacterium in the form of rods that are quite short that resembles elongated cocci. Next is Diplobacilli, which are rods in pairs, while Streptobacilli are rods in chains. Okay? Diplo meaning 2, so meaning pair, rods in pair, while strepto are in chains. So, pleomorphism. Pleomorphism is the ability to exist in a variety of shapes. Okay? Number 10 is, again, we're going to define some terms. So first is obligate aerobe. So what is obligate aerobe? Obligate aerobes are the ones that require an atmosphere containing molecular oxygen in concentrations comparable to that found in room air in order for them to grow and multiply. So next is microaerophile. So microaerophiles also require oxygen for multiplication, but in concentrations lower than that are found in room air. Okay. So next is facultative anaerobe. These are anaerobe that are capable of surviving in either the presence or the absence of oxygen. So meaning they can survive even with or without oxygen. So next is aerotolerant anaerobe. Aerotolerant anaerobe does not require oxygen at all. It grows better in the absence of oxygen, but can survive in atmospheres containing molecular oxygen such as air and carbon dioxide incubator. Next is Obligate anaerobe. Obligate anaerobe is an anaerobe that can grow only in an anaerobic environment, capnophile. Okay, so in the next item, we're going to state the key differences among rickettsias, chlamydias, and mycoplasmas. So, chlamydia is a group of gram-negative bacteria and obligate intracellular parasite that transmits from person to person. Okay? Rickettsia, on the other side, is a group of gram-negative bacteria and obligate intracellular parasites that transmit by arthropod vectors. So they differ in terms of transmission, okay? Chlamydia transmits from person to person, while rickettsia transmits through arthropod vectors. Another key difference from chlamydia and rickettsia is that chlamydia cannot produce ATP or the adenosine triphosphate, while rickettsia they can produce some adenosine triphosphate. Mycoplasmas is a genus of bacteria that lack cell wall around their cell membranes. They do not have cell walls. Okay? Rickettsias and chlamydias are similar in the sense of being obligate intracellular organisms that can only survive within the cell hosts. But conversely, both differ in their mode of transmission. Okay? Mycoplasmas are distinct in their kind, as it is pleomorphic organism that doesn't possess a cell wall. Pleomorphic meaning they can be in a variety of shape. Okay, because mycoplasmas do not have cell walls. Next is we're going to identify several important bacterial diseases of humans. So we have here pulmonary tuberculosis, diphtheria, third, cholera, fourth, leprosy, fifth, pertussis, sixth is tetanus, seventh is plague, eighth, gonorrhea, ninth, syphilis, and 10th is salmonellosis. Okay, so these are several important bacterial diseases of humans, meaning they are caused by bacteria. Number 13, we're going to state several ways in which archaea differ from bacteria. So archaea vary widely in shape. Some are coxy, some are bacilli, and others form long filaments. Many, but not all, archaea are extremophiles in the sense that they live in extreme environments, such as extremely acidic, alkaline, hot, cold, or salty environments, or environments where there is extremely high pressure. Some live at the bottom of the ocean, in and near thermal vents, where in addition to heat and salinity, there is extreme pressure. So, archaea differ from bacteria in numerous ways. So, plasma membranes are made of different kinds of lipids. In archaea, RNA and ribosomal proteins more like those of eukaryotes. And archaea are mostly anaerobic. Okay? Unlike bacteria, archaea cell walls do not contain peptidoglycan. They are covered in protein layered called capsule. Archaea lives in more extreme environment than bacteria. And archaea have more complex RNA polymerases than bacteria. And archaea do not use glycolysis pathway to break down glucose. Okay? So that's all for chapter 4, and see you in my next video.